This program demonstrates some of the things that can be done with DNA in what we call molecular biology and genetic engineering. It had long been known that DNA carries the genetic information, but it was not until Watson and Crick's discoveries in 1953 that the importance of the DNA double helix was realized. Later, the processes of protein synthesis and DNA replication were unraveled. By the early 1980s, some biochemists, who were then starting to call themselves molecular biologists, were beginning to find ways of manipulating DNA. This meant actually altering the genetic material of cells. The manipulations were carried out using enzymes that act on DNA. Soon it became possible to determine the sequence of bases in DNA, the genetic message itself. In this program, you will see how a long, double-stranded molecule of DNA can be cut using restriction enzymes, how the lengths of the pieces can be measured by electrophoresis, how a length of DNA can be amplified using a special reaction called the polymerase chain reaction, and how a piece of human DNA, in other words a gene, may be cloned by inserting it into a bacterial cell. Most research involving DNA manipulation is carried out on the micro scale, using microliters of liquid rather than milliliters or liters. This involves using specialized equipment like this. There are a number of reasons for working on the micro scale, one of which is that many of the reagents used are very expensive. This is a micropipette of the type commonly used in biochemical work with DNA. It has an adjustable micrometer plunger arrangement that allows it to deliver any volume between 10 and 200 microliters. The micropipette is used with a plastic disposable tip. Unlike glass, water doesn't cling to plastic, so we don't have to worry about droplets on the outside or about draining times. The tips we are using have been sterilized in an autoclave. This is because many microscale experiments must be performed under sterile conditions. We don't want bacterial DNA or enzymes contaminating the experiments. After use, the tip must be discarded into an appropriate container. This is particularly important if it has been used to pipette radioactive materials, bacteria or viruses, all of which are often used in molecular biology. We need other microscale equipment to go with the micropipettes. These are plastic micro-test tubes and a microcentrifuge. This is just about all the equipment we need to start doing molecular biology work on a micro scale. The first experiment we're going to see shows how to cut a double-stranded DNA molecule using a restriction enzyme. In order to explain how this works, we need to look very closely at a short section of DNA. It will be easier to see what is happening if we straighten the double helix out. The experiment uses a restriction enzyme extracted from bacteria to cut the DNA sequence in a specific place. A different restriction enzyme would cut the DNA in a different place, but each cuts at a specific sequence of bases in double-stranded DNA. The enzyme we're going to use is called HINF1. It comes from the bacterium Haemophilus influenzae. Note that the cut is not at the same place in each DNA strand, so that when the strands are pulled apart, they leave an overhang at each end. Here is how we carry out the cutting in the laboratory. A few microliters of a solution containing DNA is placed in a little plastic test tube. This contains about 50 nanograms of DNA. Ten microliters of a buffer solution is added to keep the pH constant. Now for the restriction enzyme. 1 microliter is all that is required. When this has been added, the tube is closed and the contents mixed briefly using a whirly mixer. A quick spin in the centrifuge brings all the contents to the bottom of the tube. This is then transferred to a water bath, where it is incubated at 37 degrees centigrade for 10 minutes. This allows the reaction to take place. At the end of the incubation period, we add a little of a compound called EDTA, which stops the enzyme reaction. Then we add some blue dye. This will act as a convenient marker so that we can see what we're doing in the next part of the experiment, which is to analyze the DNA by electrophoresis. This will confirm that the DNA has indeed been cut, and will also tell us how long the segments of DNA are. Electrophoresis is a gentle way of separating or analysing biological molecules such as DNA and proteins. It works by separating molecules from each other on the basis of charge. Positively charged molecules move towards the cathode in an electric field, and negatively charged ones move towards the anode. In addition, molecules with a high charge move more rapidly than those with a low charge. Size is important too. Large molecules move more slowly than small ones. Now, let's look at that last sequence again. See if you can spot what is happening. Imagine that this is a mixture of molecules of different sizes and different charges. We will now demonstrate the experiment using this electrophoresis apparatus and an agarose gel. We use a gel because we want to separate not only by charge but also by size. The gel forms a sort of sieve that lets small molecules of DNA move more rapidly than large ones. Agar is a polysaccharide obtained from seaweed, which when dissolved in water forms a gel. Agarose is a highly purified form of agar. The gel is made by putting a measured amount of agarose powder into a test tube and adding buffer. This mixture is heated in a boiling water bath until all the powder has dissolved. The liquid gel is then poured into a flat mould and allowed to cool. There is a plastic comb called a former standing over the dish at one end, with the teeth of the comb sticking into the gel. When the gel has set, this is removed. The teeth leave slots in the gel into which the samples will be placed. The experiment is performed using this Perspex electrophoresis tank. It has a reservoir at each end for buffer solution and a raised central section on which the gel is placed. There is a platinum electrode at each end of the tank. The red spot identifies this one as the positive. If we look closely, we can see the thin platinum wire electrode embedded in the Perspex. The first step in the experimental procedure is to fill the tank with buffer solution. The slab of gel can now be placed in position. First, we remove the sticky tape that held the gel in the mould while it was setting. It can then be put into the electrophoresis tank. In a moment we will put the DNA samples in and start the current, but before that we will look at some diagrams of how the electrophoretic separation works. Remember that DNA, deoxyribonucleic acid, is negatively charged because of all the phosphate groups on it. For every base there is one phosphate and therefore one negative charge. Consequently, large DNA molecules have a higher charge than smaller ones. In electrophoresis, all will move towards the positive electrode. When we carry out electrophoresis in an agarose gel, the small DNA molecules can move rapidly, while larger DNA molecules are held back by the gel, which acts rather like a sieve. So we get a separation that is based on size. Small molecules go a long way in the gel, medium-sized molecules a moderate way, and large molecules only a short distance. This is a very convenient way of finding out how many base pairs there are in a piece of DNA, in other words, how long it is. The experiment takes about an hour to run and is very sensitive. We can work with 1 microgram of DNA, or even less. Now let's see the experiment. These are the samples of DNA we're going to run in the gel. Each sample is carefully pipetted through the buffer into a slot in the gel. A steady hand is needed at this stage. When all the samples have been loaded into the gel, the lid is placed on the electrophoresis tank. It is designed so that the electrodes cannot be touched when the current is on. After connecting the positive and negative wires to the power source, we switch the unit on and set the voltage to 100 volts. After an hour or so, the electrophoresis is complete. You can see that the blue marker dye, which moves faster than any of the DNA molecules, has moved quite a long way. However, you can't see the DNA itself. We need to make it visible by staining with a dye, called ethidium bromide. This fluoresces when exposed to ultraviolet light. Now that the electrophoresis is finished, we can switch off the current and remove the lid. The next step is to stain the gel with ethidium bromide so that we can see the bands of DNA. After staining, the gel is placed on an ultraviolet light source which has a camera mounted above it. It's very important to wear a visor because ultraviolet light causes serious damage to skin. When we turn the room lights off, we can see the DNA bands. This is the positive end of the gel, so these are the smaller molecules. This track is of a sample of DNA that has molecules of different sizes. It contains the DNA that was cut using the restriction enzyme. You can see that several pieces have been produced. When a cell divides, its DNA must be duplicated. Each strand of the DNA double helix acts as a template for making a new strand. Each daughter molecule of DNA contains one parental strand and one newly made strand. This process is called semi-conservative replication of DNA. The main enzyme involved is DNA polymerase. This enzyme can take a single-stranded piece of DNA and, with a little bit of primer, make a double helix. It does this by adding new base units that are complementary to those in the original strand. In this way, a new double helix is formed. This suggested a way in which specific bits of DNA could be amplified. This reaction, which is carried out in the laboratory, is called the polymerase chain reaction. Using it, we can select a section of DNA from a long length and amplify it 1,000,000 or more times. You might ask why anyone would want to amplify a piece of DNA 1,000,000 times. Well, there are several reasons. One is to isolate a gene that we might want to place in a bacterial cell. One example could be the gene for human insulin. You might want to detect a gene, perhaps in an individual carrying a mutation, such as sickle cell anemia or phenylketonuria. The band of mutated DNA might be detected on an electrophoresis gel. You could start with a very small amount of DNA, say that contained in just a single cell. We are now going to see how the polymerase chain reaction is carried out. At the end, the products will be analysed by electrophoresis on an agarose gel. The starting point is having short lengths of DNA called primers. These are manufactured chemically on a DNA synthesizer, a June machine, like this one. These 4 bases will be added together in a computer-controlled sequence to make up the primer. The synthesizer can be programmed to produce primers that match up with specific sequences in some DNA from a cell. We need 2 primers, one for each DNA strand, that between them bracket the section of DNA we wish to amplify. The other thing we need is the DNA polymerase enzyme itself, Taq polymerase, because we're going to make more DNA. We will go through one cycle of the process slowly, so that you can see what is happening. This is the piece of double-stranded DNA we want to amplify. The gauge on the right will show the changes in temperature that are an essential part of the procedure. First, we raise the temperature to nearly boiling. This separates the 2 strands. Now we add the primers, the short bits of DNA, and cool slowly to 56 degrees. The primers find the stretches of DNA to which they are complementary and anneal to them. Next we add the DNA polymerase, represented here by the green circles. Remember that in order to make more DNA, this needs single-stranded DNA plus a primer, which is exactly what we have here. The polymerase operates best at an intermediate temperature, so now we heat the mixture to 73 degrees. The enzyme creates a second complementary strand of DNA along each of the single strands. If these temperatures seem rather high for living organisms, we'll see why they are used in a minute. We now have 2 lengths of double-stranded DNA, where we had 1 before. The whole process is then repeated. Heating to 95 degrees separates the 2 double strands into 4 single ones. We don't need to add more primers because they were originally present in great excess. So when we cool the mixture, spare primers will anneal to each of the 4 strands. The large number of primers also makes sure that the original strands are unlikely to find each other again. They are always more likely to pair up with a primer. We don't need to add any more DNA polymerase either, so when we reheat to 73 degrees, this starts working again automatically. The polymerase we're using is not adversely affected by high temperatures, since it originates from a bacterium whose natural habitat is hot springs. This remarkable enzyme survives heating to 98 degrees and works quite effectively at 73 degrees, so we don't have to add new enzyme at each cycle. Now we have created 4 double strands of DNA from the original one. We can continue into the next cycle, heating to separate the strands, cooling to allow the primers to anneal, reheating to encourage the action of the DNA polymerase. So now we have 8 double strands of DNA. The next cycle will produce 16, the next 32, and so on. 30 or 40 cycles will produce millions of copies of the original length of double-stranded DNA. Now we are going to see this happening in the lab. This piece of apparatus is called a thermocycler. It has heating blocks, the temperature of which can be precisely controlled. The sample tube is placed in one of the blocks, and the appropriate cycle of time and temperature is set on the computer keypad. This tube contains the heat-resistant DNA polymerase. It is the last active ingredient to be added to the sample. We then fill up the tube with some light oil. This floats on the top of the other contents. It stops evaporation and ensures good heat transfer without affecting the procedure. It will be removed at the end. The computer can now be instructed to run the appropriate program. At the start, the strands of DNA are separated by raising the temperature of the block to nearly boiling. Then the temperature will be lowered to allow the primers to anneal with the separated strands. 56 degrees for a minute or two is long enough for this to take place. The temperature is then raised to the point, 73 degrees, at which the DNA polymerase works most efficiently. It will add several thousand bases in a few minutes, a remarkable enzyme. That is one cycle completed. Over the next few hours, the machine will run through 30 cycles automatically. 95 degrees, 56 degrees, 73 degrees, and so on, until the program is complete. Now we take the tube out of the thermoblock and remove the oil layer. The next step is to analyze the sample by electrophoresis to see if any DNA has been amplified. We don't need all the sample for the analysis, so we take a few microliters off into another micro test tube. This is all we need to carry out the electrophoresis test. To make the sample visible on the agarose gel, a blue dye is added. The liquid is sucked back into the pipette and expelled again a few times to ensure that the dye and the sample are thoroughly mixed. You saw the electrophoresis procedure earlier. The samples are pipetted into slots in the agarose gel and the voltage is set at 100. After staining with ethidium bromide, the gel can be analysed on the ultraviolet light box. The fluorescent bands show where the DNA is. The small band here is the one that has resulted from the polymerase chain reaction. Starting from nanograms or less of DNA, we now have enough to be visible on a gel, a few micrograms. Although this may not seem very much, it is quite enough for a DNA cloning experiment. The band containing the DNA can easily be cut out of the gel with a scalpel blade and the DNA extracted from it. Cloning DNA means inserting a piece of DNA into a cell, often a bacterial cell, and then getting that cell to grow and divide to produce a clone of identical individuals, all containing replicas of this piece of DNA. Each of the colonies of bacteria you can see growing on this Petri dish is a clone of identical cells. These have been derived from a single cell by repeated division. We often want our clone of bacterial cells to express the inserted gene, that is, to produce the protein encoded in the DNA. This is a way of producing large amounts of proteins for commercial or medical use. Examples of proteins now produced in this way are human insulin and human growth hormone. The technique of cloning used daily in molecular biology labs is called recombinant DNA technology. This is because the gene in question is usually combined with a small circle of bacterial DNA called a plasmid. This is an electron micrograph of some plasmids. You can see they are composed of short lengths of DNA. The modified or recombinant plasmids are reinserted into the bacterial cells. This is an easy way of inserting a bit of DNA into bacteria and getting it expressed. Now we're going to see how cloning can be carried out in the laboratory. Only the most important steps will be shown. Suppose we want to put the gene for human insulin into bacteria. We will need some plasmids, some bacterial cells, nutrient medium, and of course the gene itself, the piece of DNA. First of all, plasmids. Where do we get them from? Plasmids are small circles of bacterial DNA, a few thousand base pairs long, that are separate from the main chromosome. Each plasmid carries a few genes, often the genes for resistance to antibiotics. Plasmids can be transferred from one bacterial cell to another. This is how resistance to antibiotics can spread. In the laboratory, we cut open the plasmid circle of DNA using a restriction enzyme. You have already seen how this is done. Now we insert our gene, say the one for human insulin, and put the closed circles of DNA back into bacteria. When the bacteria divide, they will form a clone of cells, all producing insulin. This incubator contains cells of the common gut bacterium, Escherichia coli, growing in a nutrient medium. They are a rather fastidious strain of cells and cannot survive long in the absence of the special growth medium. There is no danger of their escaping from the laboratory and spreading unwanted genes in the wild. These cells contain plasmids. When we have enough bacterial cells, they are spun down in a centrifuge to separate them from the medium in which they were grown. Next, the cell walls will be digested away with lysozyme. You may know that Sir Alexander Fleming first identified the enzyme lysozyme in human tears. After the mixture of cell contents has been centrifuged to remove any fragments of cell wall, we carry out ultracentrifugation. The pink colour is due to ethidium bromide, which has been added to make the plasmid DNA visible at the end of the procedure. The tubes are loaded into an ultracentrifuge, where the plasmids are purified on a density gradient of cesium chloride. The ultracentrifuge is run at high speed overnight. This is called density gradient ultracentrifugation. Next morning, the tubes containing the plasmids are carefully removed. The bands of DNA are clearly visible. The bottom band is the one we're interested in, as it contains the plasmid DNA. To remove this, the tube must first be clamped securely. We have to be very careful at this stage not to disturb the contents. Because the tube is made of plastic, it's quite easy to remove the band containing the plasmid DNA. First, we make a little hole in the top of the tube to let air in. This will make it much easier to extract the band at the bottom. Now insert the needle from a 2 ml syringe into the tube just below the band we want. We then simply suck the liquid containing the plasmid DNA into the syringe. Following safe disposal of the needle, the liquid is transferred to a test tube. We now add 70% ethanol. This will make it easier to remove the ethidium bromide and recover the DNA, which is precipitated by cold ethanol. The tube is sealed with parafilm and put on dry ice. This precipitates the plasmid DNA so that we can recover it by centrifugation. After the tubes have been centrifuged, the liquid is carefully poured off, leaving the plasmid DNA behind. The purity of the DNA is usually checked at this stage by electrophoresis. The band formed by the plasmid is readily visible on the electrophoresis gel. Now we are ready to insert the gene we want to clone into the plasmid circles. We're going to cut the circles open with a restriction enzyme, insert our gene, and then put the plasmids back into receptive bacterial cells. You will remember from part 1 of this video that when restriction enzymes cut double-stranded DNA, they leave overhanging ends. If our gene, the piece of DNA we wish to insert, has identical overhanging ends, it will easily fit into the plasmid circle. The plasmid is cut open with the restriction enzyme. The gene we want to insert has been cut in the same way. Therefore, the overhanging ends match up and the gene fits into the circle. The overhanging ends are sometimes called sticky ends. They match up because the base pairs are complementary to one another. Now we are ready to insert this modified plasmid into the bacterial cells. The first step is to centrifuge the cultured bacteria. You can see the cloudy suspension in the test tubes. The centrifuge is run at 3,000 revolutions per minute for 10 minutes. When the tubes are removed, the bacterial cells are clearly visible as a pellet at the bottom of the plastic tube. The liquid is carefully poured off and the pellet placed on ice. Calcium chloride solution is added to make the cells receptive to plasmids entering them. We say they are competent. The tube is tapped gently to resuspend the bacterial cells. They need to be treated with care at this stage because they are fragile. Now we add the plasmid which contains the gene we wish to clone. To make the cells more receptive to the plasmids, we need to give them a brief temperature shock. So they are placed in a water bath at 41 degrees. After a few minutes, the tube is put back onto the ice and left to stand for a while. Some fresh nutrient broth is added to get the cells growing again. Now, of course, they contain the modified DNA plasmids. At this stage, we need to increase the number of cells by growing them on agar. We put a few drops of the transformed culture onto each Petri dish. And spread them over the surface of the agar. The plates are put in an incubator overnight so that the bacteria can grow. The temperature for optimal growth is 37 degrees. Next day we take the plates out. The individual cells will all have grown into colonies. The final step is to select the colonies we want. Now when we put the plasmids into bacteria, the process is not very efficient. Only a proportion of the bacterial cells will take up a plasmid. We need to select those cells that have a plasmid in them. We do this by using the antibiotic resistance marker, The cells containing a plasmid are resistant to the antibiotic ampicillin. We grow the bacteria on agar plates containing ampicillin, so only those cells with a plasmid in them survive. There is a problem with the plasmids too. Most of them will not have taken up the extra gene, the circle's just reclosed, so only a few will have the gene we inserted. We need to find the bacterial cells that contain a plasmid with our added gene. We use a cunning plan to pick out the colonies derived from these cells. As you will see, they will reveal themselves by their colour. When we used the restriction enzyme to cut open the plasmid circle, we selected one that cuts in the middle of one of its genes. The gene we selected is that coding for a particular enzyme, lactase. So when we cut the circle and insert our gene, this will make the split gene inoperative. This means that any bacteria containing plasmids with the inserted gene can no longer make the enzyme. Those without an insert can still make it. So how can we detect which colonies can still make this enzyme and which cannot? The secret is to grow the colonies on agar containing an artificial substrate of the lactase enzyme, one that turns blue if the enzyme is present. You can see that some of the colonies on this plate are blue and some are white. The white ones, those that cannot produce the lactose splitting enzyme, are the ones we want, because they must have our gene inserted into their plasmids. The final step is simply to pick these colonies off the agar plate and grow up cultures from them. These cultures should be producing human insulin, if that's the gene that we inserted. A fermenter like this one can produce millions of copies from just a few cells. This would enable human insulin, or in principle any protein, to be produced in commercial quantities. What you've seen in this program is only a little of what constitutes molecular biology today. You haven't seen DNA sequencing or cloning using bacteriophages. Cloning is now also done in cells other than bacteria, such as yeasts. Putting genes into human cells is not all that far off as a medical treatment. In the future, this will enable individuals with genetic deficiencies to be cured simply by giving them modified genes. Molecular biology methods allow us to isolate and modify the genes required for such treatments. Both the polymerase chain reaction and cloning allow genes to be selected and amplified. In this program, you have seen some of the ways in which DNA can be manipulated, cut, analysed by electrophoresis, amplified, and cloned into bacterial cells. The tools for doing this are the enzymes that deal with DNA in cells. You will have noticed that most of the operations are carried out on the micro scale and are actually rather simple. The methods of analysis have to be sensitive in order to cope with the tiny amounts of DNA to be analysed. Almost anything can now be done with DNA in the laboratory. The key is to plan the experiments that further our knowledge and lead to a better understanding
This is an aerial view of a field of sugar beet. It should be full of healthy green plants, but it's been infected by a virus. Where the viruses have attacked the plants, the leaves have turned yellow, making the crop useless. A different virus has attacked these cattle, producing the terrible symptoms of foot and mouth disease, great suffering for the animals, and possible financial ruin for the farmer. Chickenpox, mumps, measles, even the common cold, are all caused by viruses. Fortunately, their effects are well known, and in most cases not too severe. But the effects of a virus can be devastating, as the concern and publicity about AIDS show only too clearly. Preventing the spread of viruses is not easy, although in some cases it has been done successfully. In this field, for example, the right-hand half of the crop was sprayed to kill the insects which transmit the virus, hence the healthy green plants. The spread of the polio virus is being controlled by the polio vaccine, a safe and effective way of building up resistance to the virus in the population. And vaccination has totally eliminated the smallpox virus, which used to be a major cause of suffering and death. The economic, social and medical consequences of virus infection are obviously far-reaching. So what are viruses? Viruses are very tiny particles, 400 or 500 times smaller than a red blood cell, which can only be seen in an electron microscope. They are made up of nucleic acid and protein molecules. The nucleic acid is either DNA or RNA, depending on the virus, and it's surrounded by a protective coat of protein. The protein coat usually consists of lots of identical subunits, but the way in which these are arranged is characteristic of different viruses. In the tobacco mosaic virus, for example, a virus which infects tobacco plants, the protein subunits are built around a coiled RNA molecule, forming a tube. This is what it looks like when it's magnified several thousand times in the electron microscope. In contrast, another virus, called adenovirus, which causes respiratory infections in young children, is nearly spherical. This virus contains DNA, and the protein coat packs around it to form a geometrical figure with 20 faces, an icosahedron. The protein subunits of the adenovirus show up well in the electron microscope. It's even possible to see that this virus has some extra spikes of protein radiating out from the coat. These are involved in attaching the virus to cells. Some viruses, like the virus that causes AIDS, have a surrounding membrane or envelope. This is made up of lipids and proteins, with the proteins projecting through the membrane. The electron microscope shows that the human influenza virus also has this type of structure. A virus that looks entirely different from the ones you've seen so far is the T2 bacteriophage. This is one of a family of viruses which attack Escherichia coli, a rod-shaped bacterium that lives in the human gut. The DNA of this virus has its protein coat arranged in the form of a cylinder with flat sides and pointed ends, but it also has a tail section. This is used during virus infection. It helps to anchor the virus to the wall of the bacterium. Viruses flourish by making more viruses, but they can't do this on their own. Making new viruses involves making more nucleic acid, making more protein subunits to coat it, and then assembling the nucleic acid and the proteins correctly. And even the most complex virus doesn't have the biochemical machinery for doing this: enzymes, ribosomes, and so on. A virus can only make more viruses by invading a host cell. For some viruses, the host is a bacterium. For others, it's a plant or animal cell. In all cases, the life cycle of the virus is essentially the same. The nucleic acid from the virus enters the host cell. This nucleic acid is the genetic material of the virus. It contains the code for the proteins of the virus coat and the other proteins necessary for the new viruses to be manufactured and assembled. When it gets into the host cell, the protein synthesizing machinery of the cell is taken over to make these proteins, enough for several hundred new virus particles. More of the virus nucleic acid is made as well, and the new virus particles are then assembled and released from the cell. Viruses which are surrounded by a membrane or envelope, like the AIDS virus, are assembled at the plasma membrane of the host cell and released by budding. So each new virus particle comes away coated with a little piece of the plasma membrane of the host cell. This electron micrograph shows a number of new AIDS viruses budding from a cell. With other host cells, the assembly and release of viruses often cause the cell to burst. A suspension of bacterial cells, for example, has a cloudy appearance, the result of light scattering by the cells. But if a virus is added to the suspension, the cloudiness will clear as the cells are infected and burst open. This agar plate has a uniform growth of bacterial cells on it. If the plate was exposed to a virus, wherever the viral nucleic acid entered a bacterial cell and took over, more viruses would be produced, taking over more cells, until eventually all the cells in that region burst, leaving clear spots. These clear spots are known as plaques, and they provide a very useful way of detecting virus infection in bacteria. Each plaque originally came from one virus particle. Staining the remaining bacteria with a dye makes it easier to count plaques. For a virus to enter a host cell, it must first attach itself to the surface of the cell. It can only do this if the surface has specific receptors for that virus. Consequently, each virus has a very restricted range of host cells. Plant viruses never attack animal cells, for example, and most of the viruses that attack domestic animals can't harm humans. The nucleic acid from viruses gets into cells in different ways. Animal viruses generally enter their host cells intact, and it's only when the virus is inside the cell that its coat is removed and the nucleic acid can start taking over the cell's metabolic machinery. On the other hand, bacterial viruses, like the T2 bacteriophage, usually inject their nucleic acid into the bacterial cell, leaving the protein coat outside. It was the T2 bacteriophage that was used by Hershey and Chase in their experiments to show that DNA, not protein, contains the genetic information. When Alfred Hershey and Martha Chase did their experiments nearly 40 years ago, biologists were not sure how genetic information was carried from one generation to the next. It was known that in eukaryotic cells, the genes are located in the chromosomes in the nucleus. It was also known that chromosomes are made up of DNA and protein. So either could have been responsible for passing on the genetic information. In the case of DNA, its double helix structure hadn't been worked out. It was known that DNA consisted of long chains of nucleotides, but it was thought that the simple repeating structure of sugar, base and phosphate was much too simple to carry the complex genetic message. Proteins, on the other hand, obviously were complex structures, and they occurred in many different shapes and sizes, and with a variety of different properties. So perhaps it was the proteins closely associated with DNA that passed on the genetic message. Hershey and Chase were among the first to answer this question through their experiments with the T2 bacteriophage. They chose this particular virus because they knew that it contained only DNA and protein, and because it readily reproduced itself in a bacterial cell. First of all, they prepared some radioactively labeled T2 bacteriophages. To do this, they grew bacteria in a medium containing radioactive phosphorus, as is happening here. When the bacteria took in nutrients from the medium, they also took in the radioactive phosphorus, which became incorporated into many different molecules in the cells. The suspension of bacteria was then infected with the T2 bacteriophage. To make new virus particles inside the bacterial cells, materials from the cells have to be used. So some of the radioactive phosphorus was passed on to the virus particles. When the virus infection was complete, and the bacterial cells were full of new viruses, they burst, and the cloudy suspension became clear. The very tiny virus particles were separated from the fragments of the bacterial cells by centrifuging. The cell fragments were pulled to the bottom of the tube, leaving the virus particles in the supernatant. This was centrifuged at higher speeds to purify the virus particles. This is the concentrated suspension of purified viruses, and a Geiger counter confirms that the viruses are now labelled with radioactive phosphorus. It's the radiation given off by the radioactive atoms that makes the counter click. Now, a T2 bacteriophage only contains DNA and proteins, and there is no phosphorus in proteins. So the radioactive phosphorus must have been in the DNA part of the virus, in the repeating units of base, sugar, phosphate. Making the DNA of the virus radioactive allowed Hershey and Chase to follow it wherever it went, and they did an experiment to see what happened to it when the virus infected bacteria. They took a suspension of bacteria that had no radioactivity in it, and they added the radioactive virus. They left the mixture for a few minutes to allow the viruses time to attach themselves to the bacterial cells and pass materials into them. If the virus DNA had entered the cells, the radioactivity should now have been inside the cells. To check this, the virus material remaining on the outside of the cells had to be removed. This was done by putting the suspension of cells and viruses in a blender and rotating the blades vigorously for a few minutes. This treatment broke all the remaining virus material off the surface of the cells, and when the mixture was spun in a centrifuge at low speed, the bacterial cells were pulled to the bottom of the tube, leaving in the supernatant the virus material that had not entered the cells. If the DNA from the virus had been passed to the cells during infection, the pellet of cells should have been radioactive. and the Geiger counter showed that it was. To see if there was any radioactivity in the virus material left outside the cells, the supernatant was centrifuged to sediment the material. This needed to be done at a much higher speed because the virus fragments are very much smaller than the bacterial cells. After centrifuging, the material that had sedimented was tested for radioactivity. It hardly contained any. This experiment showed that when the virus attaches itself to the bacterial cell, its DNA passes into the cell. But it was still possible that some protein passed in too. This wouldn't have been detected because it wasn't carrying a radioactive label. So Hershey and Chase did another experiment to see if any protein from the virus entered the bacterial cell. The experiment was exactly like the one you've just seen, but with one important difference. Instead of labeling the DNA part of the virus with radioactivity, they labeled the proteins, using radioactive sulfur. They could do this because proteins contain sulfur. It's in the amino acid cysteine, for example. DNA doesn't contain sulfur. So, if bacteria were grown in a medium containing radioactive sulfur and were then infected with the T2 bacteriophage, new viruses were formed which had radioactive protein coats. The purified radioactive viruses were then used to infect non-radioactive bacteria. After allowing time for the viruses to pass material to the cells, the protein coats of the viruses were broken off the outsides of the cells in a blender. The cells and protein coats were then collected separately by centrifugation. When the pellets were checked for radioactivity, the Geiger counter showed that the bacterial cells contained very little radioactivity. The protein coats, on the other hand, were highly radioactive. This showed that no virus protein had entered the bacterial cell during infection, and confirmed that the DNA of the T2 bacteriophage must be the carrier of the genetic information. Although Hershey and Chase's experiments only proved this for viruses, in the last 40 years many other experiments have been done which show that in all living things, nucleic acid is the carrier of genetic information. In any living cell, there are thousands of different proteins, some forming part of the cell's structure, some responsible for its complex functions. All proteins are chains of amino acids, and the chains of each type of protein have a unique three-dimensional shape. It's the sequence of amino acids in a protein chain that determines how that chain will coil and fold up. And for every type of protein, that sequence is different. To keep functioning properly, a cell must be able to replace or renew its proteins. When it's growing or dividing, it must be able to make new proteins. The information that allows the cell to do this is stored in the nucleus in coded form in the molecules of DNA. Each strand of a DNA double helix is a chain of nucleotides, Each nucleotide is made up of a base, sugar, and phosphate. The chains contain 4 different bases, either cytosine, C, guanine, G, adenine, A, or thymine, T. And it's the sequence of these bases in DNA that contains the information for the sequence of amino acids in proteins. The information is in the form of a triplet code. In other words, a sequence of 3 bases is the code for one amino acid. So, if you know the sequence of 3 bases for each amino acid, you can interpret the sequence of bases in a length of DNA as the sequence of amino acids in a protein. In this example, it would be alanine, serine, lysine, and so on. DNA stores the instructions for protein synthesis, but before the cell can act on these instructions and make proteins, a number of other things are needed. The information about protein structure, stored in the genetic code of DNA, is in the nucleus. Protein synthesis takes place on tiny organelles called ribosomes, which are in the cytoplasm. A messenger molecule is used to transfer the necessary information from nucleus to cytoplasm. The messenger molecule, messenger RNA, is made in the nucleus on a single strand of DNA, and the sequence of bases in its nucleotide chain is determined by the sequence of bases in the DNA. Wherever there's an adenine in DNA, there's a uracil, U, in messenger RNA. Where there's a thymine in DNA, there's adenine in messenger RNA. Where there's cytosine in DNA, there's guanine, and where there's guanine, cytosine. This is called base pairing, and the 2 bases which form each pair are said to be complementary. As a result of base pairing, the genetic code in DNA gives rise to a complementary code in molecules of messenger RNA, with a sequence of 3 bases, known as a codon, specifying one amino acid. It's in this form that the instructions for making a protein are passed to the ribosomes. Ribosomes are made of RNA and protein. The ribosomal RNA consists of a number of RNA molecules of different chain lengths, from several hundred to several thousand nucleotides. These molecules coil and fit together with the ribosomal proteins to form 2 irregularly shaped subunits, one large, one small. Together, these form a structure on which all the various molecules involved in protein synthesis can come together and function correctly. Amino acids are brought to the ribosome by yet another type of RNA called transfer RNA. There's at least one transfer RNA molecule for each amino acid, but they all have the same general structure. Drawn like this to show how the different parts of the nucleotide chain bond together, a transfer RNA molecule looks a bit like a clover leaf. But in 3 dimensions, it's more like a twisted hairpin. And in a simple form, it can be drawn like this. There are 2 parts of a transfer RNA molecule which are very important. At this end of the molecule, the transfer RNA attaches to its corresponding amino acid. So, if this was the transfer RNA for alanine, alanine would be attached here. If it was the transfer RNA for serine, serine would be attached here, and so on. Attaching an amino acid to its transfer RNA needs energy from ATP and specific enzymes which can recognize both the amino acid and the transfer RNA. At the other end of the transfer RNA molecule, there's a sequence of 3 bases which is specific for the amino acid it carries. In the case of alanine, these 3 bases would be CGA. For serine, they would be AGU, and for lysine, UUC. The 3 bases here make up the anticodon, and their sequence is complementary to the sequence of bases making up the codon on messenger RNA. This means that transfer RNA molecules can line up on a messenger RNA molecule, but they can only do so in a certain order. That order is determined by the order of the codons in messenger RNA, which, in turn, is determined by the sequence of bases in DNA, the genetic code. The interaction of codons and anticodons at one end of the transfer RNA molecule ensures that, at the other end, amino acids are lined up to make proteins in the order originally specified by the sequence of bases in DNA. So, the sequence of bases in a length of DNA is first copied or transcribed into messenger RNA and then translated into the sequence of amino acids in a protein. Protein synthesis begins when a messenger RNA sent out from the nucleus links up with 2 subunits of a ribosome and with the transfer RNA carrying the first amino acid of the protein sequence, a process that also needs the help of various initiation factors, soluble proteins from the cytoplasm. In eukaryotic cells, the first amino acid brought to the ribosome is always methionine. This doesn't mean that all protein chains start with methionine. This methionine is usually removed once the protein chain has been made, but it does seem to be necessary to get the process going. The codon for methionine is AUG. This codon is known as the initiation codon, and it marks the start of a protein sequence. When it engages with the anticodon on the transfer RNA for methionine, protein synthesis is set in motion. Now, a ribosome has 2 sites where a transfer RNA molecule can bind. So, 2 transfer RNA molecules can be associated with a ribosome at any one time. These sites are called the P site and the A site. Abbreviations for peptidyl and aminoacyl sites. When protein synthesis is set in motion, the transfer RNA carrying methionine is located at the P site, which is adjacent to the initiation codon AUG. The next codon on the messenger RNA, here it's GCU, is adjacent to the A site. The anticodon to GCU is CGA. Remember, there's complementary base pairing. And the transfer RNA molecule with that anticodon is the one for alanine. So that's the transfer RNA which comes and binds here, bringing alanine alongside methionine. A peptide bond forms between them, joining the first 2 amino acids of the protein chain. In the process, the bond between methionine and its transfer RNA is broken, and it's the breaking of this bond that supplies the energy to form the peptide bond. 2 amino acids are now joined together, but Before the next amino acid can be added, two things happen. The transfer RNA, which no longer has an amino acid attached to it, leaves the ribosome so that the P site is vacant, and the whole ribosome moves along the messenger RNA until the transfer RNA, which has the growing protein chain attached to it, occupies the P site, and the A site is adjacent to the next codon on the messenger RNA. In this example, it's UCA. Now the process is repeated. The transfer RNA with the anticodon to UCA binds to the A site, bringing its amino acid, serine, alongside alanine. Another peptide bond is formed. Then the transfer RNA, which no longer has an amino acid attached, leaves the P site, and the whole ribosome moves along until the transfer RNA with the growing peptide chain occupies the P site, and the A site is adjacent to the next codon on the messenger RNA. We still don't know exactly how all this happens, but we do know that it involves a number of other molecules beside those shown here. These are soluble proteins from the cytoplasm called elongation factors. The process also needs a supply of energy. Watch the sequence again while the next amino acid is added on. The transfer RNA binds to the A site so that its amino acid is lined up with the protein chain. A peptide bond is formed. The previous transfer RNA leaves the ribosome, and the whole ribosome moves along. This sequence of events is repeated over and over again until the protein chain has reached its full length. Each time, one more amino acid is added to the protein chain, and the ribosome moves a bit further along the messenger RNA. In the cell, this process happens very quickly. To make a small protein, one with about 100 amino acids, takes less than 10 seconds. At the end of the information sequence on the messenger RNA, the ribosome meets a termination codon, which may be UAG, UGA, or, like this one, UAA. None of the transfer RNA molecules in the cell has an anticodon to these, so no more amino acids can be added to the chain. Instead, a release factor, another soluble protein from the cytoplasm, binds to the ribosome. This causes the protein chain to be released into the cytoplasm and, of course, ends protein synthesis on that particular ribosome. Usually, though, a number of ribosomes thread their way along a length of messenger RNA at the same time. You can see the ribosomes in this electron micrograph. In fact, there are so many of them clustered on each length of messenger RNA that it's very difficult to see the messenger RNA at all. Complexes like these are known as polysomes, short for polyribosomes, and they enable a number of molecules of the same protein to be produced simultaneously. By making new proteins in this way, the cell is able to grow and divide, keep its structure intact, and carry out all its complex life processes. Whenever you see this sign in a biology laboratory, you know that radioactive materials are being used. Many of them can be detected with a Geiger counter. The more clicks the counter gives, the more radiation is being given off. Radiation is given off by radioisotopes. The atoms of most chemical elements occur in several different forms or isotopes. Carbon, for example, has isotopes of mass number 12, 13, and 14. Sometimes one of the isotopes of an element is unstable and breaks down, giving off radiation. This form of the element is called a radioisotope. Isotopes which are radioactive obviously have different physical properties from non-radioactive isotopes. A higher mass number, for example, but they have exactly the same chemical properties. This makes them one of the most powerful tools available to biologists and biochemists. A living cell can't distinguish between the radioactive and non-radioactive isotopes of an element. So radioisotopes can be used to tag or label molecules. This means replacing some of the atoms in the molecule with radioactive atoms, so that the molecule can be followed wherever it goes in the cell or the organism. Its presence is detected by the radiation it gives off. And these days, very tiny amounts of radioactivity can be detected and measured very accurately. There are a number of ways of detecting radioactivity. It will blacken photographic film, for example, and this property is often used in biological experiments to produce autoradiographs. This chromatogram's been run in solvent, dried, and then sprayed with a color reagent. This revealed the position of the sugars, but doesn't show which ones are radioactive. To find this out, the chromatogram is taken into the darkroom and put next to a photographic film between two sheets of glass. It's left there for a few days, and then the film is developed to produce an autoradiograph. Wherever there was radioactivity on the paper, the film is blackened, so it's easy to tell which of these sugars is radioactive. This blackening of photographic film by radiation is also used as a safety check for people who work in laboratories where radioactive materials are used. Inside these film badges, which everyone in the laboratory must wear, there are little pieces of photographic film. At monthly intervals, the film is replaced and the old film developed. The blackness of the film is an indication of how much radiation the person has been exposed to. This approach is useful for detecting radiation in general, but it's not sufficiently accurate to give good quantitative measurements. More accurate methods for measuring radiation depend on the type of radiation being emitted. The three isotopes most commonly used in biology are P32, C14, and H3. These all emit beta radiation, streams of high energy, fast-moving electrons. One of the simplest ways of detecting beta radiation is with a Geiger counter. The Geiger-Müller tube, named after its original designers, is an ionization chamber, a gas-filled tube containing an anode and a cathode. with a high potential difference between them. When a charged particle, like an electron, passes between the two electrodes, there's a transient increase in the conductivity of the gas, which gives a pulse of electricity. This pulse is fed to a loudspeaker to give the clicking sounds, and recorded as counts. Quantities of radiation are therefore often expressed as counts per second or counts per minute. The more counts there are in a given time, the more radiation is present. But the number of counts recorded is not the same as the number of radioactive decays in the sample of isotope. This is because this type of equipment isn't very efficient at detecting every radioactive disintegration or decay that takes place near it and recording it as a count. For instance, although a radioactive substance emits radiation in all directions, only some of it will be directed towards the Geiger-Müller tube and trigger a discharge. The rest may go undetected. When one radiation particle has caused a discharge, a finite time has to elapse before another can be detected. So, at high levels of radioactivity, a significant number of radioactive disintegrations taking place will not be recorded as counts. In addition, it's almost impossible to use a Geiger counter for measuring tritium, which only gives out relatively low energy electrons. So instruments called liquid scintillation counters have been developed in order to increase the sensitivity and make the very accurate measurements of beta radiation that are necessary in modern biology experiments. You'll find these in biology and biochemistry laboratories all over the world, and the principle on which they work is very easy to understand. When certain organic chemicals are bombarded with beta particles, they emit light. So, if a radioactive compound and one of these chemicals are mixed together in a suitable solvent, the radiation given off by the radioactive compound will cause the organic chemical to give off flashes of light. A liquid scintillation counter is a machine which can count the number of light flashes, and the more light flashes there are in a sample being counted, the more radioactive compound is present. These samples are ready for counting in the machine. Each glass vial contains the radioactive compound and the special organic compound in its solvent. When the sample is taken down into the machine, the light flashes are detected by a photomultiplier tube, and each flash of light is recorded as a count. The number of counts per minute being recorded is shown here during the counting and can be printed out at the end. The photomultiplier tube in the scintillation counter responds much more rapidly than the tube in a Geiger counter. It can also detect a very high proportion of all radioactive disintegrations that take place in the sample, so even extremely low levels of radiation can be measured. Radioactive isotopes are used by biologists to study many of the cell's vital processes. One of these is protein synthesis. When a cell makes proteins, the genetic information stored in DNA is carried out into the cytoplasm by messenger RNA to the ribosomes. There, amino acids brought to the ribosomes by transfer RNA molecules are assembled into proteins. These are the red cells of the blood, the erythrocytes. In each of these mature red blood cells, there are nearly 300 million molecules of hemoglobin, the protein that carries oxygen from the lungs to the tissues. All this hemoglobin is made by an immature form of the red blood cell called a reticulocyte. Unlike the mature cell, the reticulocyte has a nucleus, so it has DNA and all the information for making proteins. This tube contains a suspension of reticulocytes from a rabbit. It's red because the cells contain hemoglobin. You can't see the individual cells, of course, they're much too small. The cytoplasm of these cells contains everything necessary for making hemoglobin: messenger RNA, ribosomes, amino acids, transfer RNA molecules, and a source of energy. So it makes a good experimental system for demonstrating protein synthesis. These reticulocytes are being kept very cold at the moment to preserve the structure of the delicate enzymes and to slow their activities right down. The demonstration is easier to do if the outer membranes are removed from the cells. This ensures that anything added to the system is added directly to the cytoplasm and the protein synthesizing system. To remove the membranes, distilled water is added to the cells. The water is drawn into the cells by osmotic pressure and bursts the cell membranes. As they burst, the cell contents are released. The fragments of cell membranes are removed by spinning the tube in a refrigerated centrifuge. The membranes pack to the bottom, so the protein synthesizing system of the cytoplasm can be gently separated into another tube. Now, protein synthesis can't be demonstrated in a system like this by weighing the amounts of protein present at different times, because any increase is far too small. But protein synthesis can be demonstrated using a radioisotope. If a radioactively labeled amino acid is added to a protein synthesizing system, it's incorporated into the growing protein chains on the ribosome. So the proteins which are made in the following minutes become radioactive. The radioactive label allows the newly synthesized protein to be detected, even if only a tiny amount is present. The appearance of radioactive protein in this system would be proof that protein synthesis was taking place. So all that's necessary is to add a radioactively labeled amino acid, take samples from the mixture during the following minutes, separate the protein from the samples, and then show that the protein is radioactive. To be sure that any radioactivity detected in this last step is from radioactive protein, the protein must be separated from the sample in a way that ensures that there are no radioactive amino acids still present. This can be done very simply. These two tubes contain some acid. If amino acids are added to the tube on the left, there's no visible change. If protein is added to the tube on the right, the protein precipitates. So, adding a mixture of amino acids and proteins to acid will make the proteins precipitate, but leave the amino acids in solution. In the same way, acid is used in the experiment to separate radioactive proteins formed during the experiment from any radioactive amino acids which may remain in the cytoplasm. Going back to the main experiment, some of the protein synthesizing system that's been kept cold is put into a water bath to bring its temperature back to 37 degrees centigrade, body temperature. To start the experiment, a small amount of a radioactive amino acid is added and the contents of the tube carefully mixed. After 5 minutes, a small sample is taken out and added to cold acid. With acid at this strength, all the proteins in the cytoplasm should be precipitated, including the enzymes, so no more protein synthesis should take place in this sample. But keeping the tube in ice is another way of ensuring that there are no further enzyme-catalyzed reactions. 5 minutes later, a second sample is taken from the system and added to acid. And this process is repeated every 10 minutes until there are 6 samples. Finally, some acid is added to the main tube to stop any further protein synthesis in the bulk of the reaction mixture. To collect the protein from this sample, remember that's the precipitate, the sample is filtered through a little piece of filter paper. It's washed well and then dried, first with ethanol, and finally with acetone. The same process is repeated for the other 5 samples. To find out if this dried protein contains any radioactivity, it must be put into a vial with some scintillation fluid and counted in a scintillation counter. Radiation from the radioactive protein will cause the organic chemicals in the scintillation fluid to give off light. This will be detected by the scintillation counter and recorded as counts per minute. Each of the 6 samples is going to be counted for 4 minutes, and once the machine has been set and started, it runs automatically, counting each sample in turn and printing out the results. It's obvious that all the samples were radioactive, and the amount of radioactivity increased with time. There's no doubt at all that the experimental system from the rabbit reticulocytes was synthesizing protein, but it wouldn't have been possible to show that without using a radioactive isotope.